So I'm Simon Mitchell. I'm a lecturer in cancer research at Brighton and Sussex Medical School. Joined in June last year, so a little over a year now. And my main research focus is on B-cell lymphomas, but that's quite a new focus for me. Before that, I was in an immunology lab. Before that, I was focusing on iron regulation. So I'm a systems biologist more broadly is what I do, computational biology. Yeah. So it was a Leukaemia UK John Goldman Fellowship, which is, it's fantastic for me. My first big grant since joining a year ago. And it lets me hire a postdoc for 2 years to pursue this project. And I'm lucky that Leukaemia UK sort of share my vision for the future of leukemia treatment and particularly lymphoma treatment in this project. Because it's quite a novel project, you know, it's combining computer science and mathematics to simulate lymphoma in the computer and try and find drug targets that way, which is probably not like any other proposal they got. But with lymphoma treatment, we've reached this stage where we haven't made progress for 10, 20 years. And so new approaches are needed, and they shared my belief in that. So that was what this project will be, to tackle diffuse large B-cell lymphoma and to really create simulations that represent individual patients. So rather than everyone getting the same treatment, I want to try and simulate patients so that we can get the right treatment to the right patient. So the project breaks down into 3 main aims. The first is to set up those personalized simulations. So take a look at data, what mutations exist in which patients, and set up simulations that represent those patients. And that's a tool for me, but also for the wider research community, because we'll make those models, those simulations available, so they'll be useful for other people. And then look at those models and try and figure out the best way to treat those virtual patients. So find the best drug for the right patient. And then the final piece of the puzzle for aim 3 is to try and identify a biomarker, something you can measure that lets you know which drug to give to which patient. So if this protein is highly expressed or this gene is highly expressed, we think they'll respond well to this drug. So that's the project, and it breaks down into roughly 3 aims over 2 years. Yeah, so along with the project, I have mentorship from Chris Pepper, who will be guiding me on this new venture, and he's wouldn't have got the award without his mentorship already, so that'll be a continuation of that. But apart from that, it's mainly work in my group, and then I've got an expanding sort of list of collaborations that I can draw on when I need to. But the project itself is mainly me and Chris Pepper, and then I've got collaborations with various members of the Haematology Research Group at the University of Sussex that I'm a member of, some of whom helped me with my mock interview, so I have them to thank for that practice. Yeah, so it's a 2-year project that I'll do at BSMS, and hopefully catalyzing more work from that, because it's really, there's this gap in your career when you start as a lecturer, your first independent position where you want to grow your group, and up until then you've been in someone else's group where you have to establish independence, test a new idea, get some funding to generate some data that lets you get more and more grants. So hopefully it's 2 years, but the beginning of many more. So actually the initial application paperwork was fairly light touch, not quite sparing for the bureaucracy that anyone who's applied for a grant will be used to, going through all these bureaucratic hurdles and filling in those forms that can be quite cumbersome. But this one was okay. This one was pretty good. I think I applied in about February and got notified of the interview in June, the beginning of June, and interviewed at the end of June, and was notified of the award in July. Before the interview, I had a mock interview and practiced with Erica Mancini, Michelle West, and Helfried Achega from the Genome Damage and Stability Center. You need to get some people from slightly outside your field to help you as well. And that was really useful. First time trying anything like that, and over Zoom, of course, as well. And yeah, my preparation was that, but also the beauty of doing these virtual interviews is you can record yourself and play it back and see how you did. So I did a number of practice interviews to just an empty Zoom room where I recorded it to the cloud and then watched it back and figured out what was good and what was bad. Yeah, the COVID situation is interesting. It's actually not a bad time to be a computational biologist, because I can connect from my laptop on my kitchen table to my server and do most of my work okay. So my productivity itself hasn't been too affected, other than the issues of working from home that we're all facing. But the project itself, we'll hire a postdoc who may need to start remotely potentially, or working from, not coming to the office too much. Us computational people, that's the downside. We're not the first people in line to get back into the office, because those labs are needed. So we might work remotely and have a few Zoom calls at the start. But overall, it shouldn't impact the progress of the project too much. We can do this sort of work from anywhere. We can call our collaborators from anywhere. So it's not a bad time to be a computational biologist. And I've got lots of my collaborators who usually do experimental work, emailing me and asking for advice because they've tried to take up some computational work during this time too.